Hey guys, it's Brandon, and my final project was based on a new way that CRISPR is being used. For those of you who are wondering what other cool things that CRISPR-related proteins could do, I'm about to go into that a little bit. So the CRISPR protein that is being used is called CRISPR-Cas13a, and it has been found to have many profound benefits in a different way than we have seen with CRISPR-Cas9 throughout this semester. This was first found by a team of researchers including a familiar name, Jennifer Doudna, who initially called this CRISPR-C2C2. They found that using this new CRISPR-Cas13a protein, they experienced what seemed to be RNase activity. So instead of targeting DNA, like we have experienced with CRISPR-Cas9, it's actually targeting single strands of RNA. And to target this RNA, they have added what they call CRISPR RNase, which is similar to what we think of when we talk about short guide RNA, and it's what allows it to target specific sequences. The very interesting thing about how this works with the CRISPR-Cas13a is what happens after it binds to its target. Just after the binding and cutting of the target occurs, something that researchers call a collateral effect happens. The CRISPR complex becomes activated and subsequently starts to cut all of the RNA strands within its proximity, whether those strands have the target sequence or not. So basically, once the target RNA strand is present, all the other RNA in that area will be cut as well by this complex. This is a very odd but exciting phenomenon because it opens the door to many potential uses in terms of diagnostic testing. So now that we know what this collateral effect is and what it does, the idea is you can place RNA linked to reporter molecules where you believe the target will be. So basically you're going to add RNA that's not necessarily the target, but RNA that will fluoresce when it's cut, so that if the target is present, when the RNA complex becomes activated, the ones that will fluoresce will also be cut. Due to this collateral effect, we will easily be able to determine that the target molecule is present in solution. And just like with CRISPR-Cas9, what actually happened was the Broad Institute kind of took over the research and took it a step further with something that they call Sherlock. Individuals from the Broad Institute of MIT and Harvard, the McGovern Institute for Brain Research at MIT, the Institute for Medical Engineering and Science at MIT, and the Wyss Institute for Biologically Inspired Engineering at Harvard University developed this method. Sherlock is an acronym that stands for Specific High Sensitivity Enzymatic Reporter Unlocking, and the process takes a single input of DNA or RNA and results in an extreme volume of RNA. So basically it's just an amplification process. The Sherlock method combined with the CRISPR-Cas13a method took what could have been a difficult process and made it something very possible. The Sherlock approach increases detectability by 1,000,000-fold, which is essential in the optimization of this CRISPR-Cas13a method. Using these methods together, they can detect viral RNA at very minimal concentrations, which means that this CRISPR can be used for diagnostic testing of so many different things. Some examples that they have talked about, which I find highly fascinating, include the detecting ability of the Zika virus in a patient's blood or urine sample within only a couple of hours. They can distinguish different types of bacteria. They can detect antibiotic-resistant genes, and they can even detect RNA that is distinctive of cancer cells. Another cool part of Doudna's research and her idea was that different variants of this CRISPR-Cas13a protein cut the RNA strand at different bases. So some complexes will cut at the adenine, whereas some will cut at the uracil. So the idea is you could have two different colors that would distinguish a positive test for two different things. So basically you could test one patient for, let's say, a Zika virus as well as a cancer-related RNA strand, and you'd see whatever color corresponds to that distinctive positive test. So you could test for several things in patients at the same time. A lot of people are saying that you can also just use this to detect DNA because you can just have that DNA turn into RNA to be targeted, and that will give you enough information about what you're looking for. So that's another approach that's really interesting, and that could be used for a lot of applications as well. Also, when we're looking forward at where this could be done and for who can get tested like this, it's very optimistic because this was designed as a paper-based test and it doesn't require refrigeration, so it can be done in places such as field hospitals, irregular hospitals. I mean, they could be used in rural clinics that lack advanced equipment. It's very cheap, and that's what makes it very appealing. Even the Sherlock approach with the amplification of the RNA and DNA segments into a high volume of RNA for easy detection, even that method is very cheap and can be used in many settings. So this new approach for diagnostic testing should be widespread in my opinion, and I believe that it's going to be what you see in the future for what we're using to detect these types of diseases. Looking forward, I'm really excited to see where this ends up going and develops into and what uses it's going to have in the future. Other than that, that's all I have for this video, and thank you guys for listening.
Created using Powtoon.